Um, if my microphone ever goes out, just wave at me or talk or start talking and I, I've been having to periodically reset it. I don't know why.
Okay, we'll let you know.
Maybe it's like gamma rays or something interfering with communications. I don't know. Anyways, thank you very much for the opportunity to speak to you today. Um, I'm going to start the talk at a high altitude and start with just talking about the brain. Let's see here. So the brain is actually the fattiest organ in the human body, which I didn't know. I never would have thought fatty brains before. Um, but it is the fattiest organ, and most of that fat is made up of lipids, membranes. But overall, the brain facts are that it consists of a lot of neurons and a lot of connections. It has over 86 billion neurons and more than 1,000 trillion connections. I can't even wrap my brain around that. Um, Information travels at high speeds, about 100 meters per second. It runs on only 20 watts of power, and you can compare that to how many kilowatts or megawatts our supercomputer here at RNL runs in order to do calculations. It's a very efficient way of doing all these calculations in this incredibly interconnected network of neurons. And again, 60% of the brain mass is actually fatty material. And the neurons that make up the connections between the brains and the synapses between them basically are involved in converting chemical signals to electrical signals, and then back and forth and so on. And it had multiple structural components. Signals come into a neuron from the dendrites. They're processed by the cell body, and the nucleus contains the genetic material necessary for the cell. The electrical signal that's being transduced is sent down the axon, and it's known as an action potential. And you can kind of see the schematic here, what's happening. And it goes on to all the other connections with other neurons. And you see that there's a large number of synapses and synaptic connections per neuron. That's why there's so many connections compared to the number of neurons. Myelin sheaths are little sections of insulator that help the action potential go faster down the axon. And at the microscopic or nanoscopic level, what happens is that when you have a connection, you have these little vesicles that pop open on what's known as the presynaptic terminal, that is going from the one neuron to the next one. These neurotransmitter molecules diffuse across about a 40 nanometer cleft, and then they're picked up by receptors. And that sends the chemical signal and then the electrical signal down the chain after that. Now, one thing that I found very surprising was this recent article in, I think it was Science, or it might be Nature, where they found that there are certain allometric rules for the number or the density of ion channels for the size of the brain of an animal in the mammalian class. So here we have the relative sizes of the brain of the different brains, going from an elephant shrew, the smallest, to a human. And what they found was, if they plotted the ratio of the density of ion channels versus the size of the cortex, it followed a line, except for humans. For some reason, humans have far fewer ion channels relative to our size compared to the other mammals. And so that's surprising, because you would think that we would have a lot more ion channels since we're so much, smarter than other mammals, right? But I guess there must be some other way of doing computation. There must be some other way of having memory processes occurring in the brain that don't rely just on ion channels. Now, the way that memory is stored in the brain, a common model for that is known as the Atkinson-Schrieffer model. And it basically has three components. You have sensory information coming into your central nervous system. You can have a process of feedback with constant rehearsal of having that memory being imprinted on your short-term memory store over and over again, and then from that be able to have a retrieval of long-term memories. And so where that happens is generally known to be right in this area right here. This is the hippocampus, this little twisted braid thing right here. And what happens is that as your your cortical part of your brain takes in information from the outside world, it gets encoded into long-term memory in this center right here in your limbic system. And then when you retrieve that, they actually have data that shows that this retrieved memories from this part then actually light up the different parts of your cortical lobes here. And to give you a kind of an anecdotal story, for whatever reason, when I was about 10 years old, I was driving, in the car with my parents, and I saw this really strange house on the street that was like mustard colored. It was very weird looking. And I thought to myself, and I don't know why I did this, but I thought to myself, I'm going to remember this house for the rest of my life. And so I just over and over and over again, I thought about the house. And this, maybe I did this for maybe 10 minutes and then I didn't think about it for a while. But I'm still, even now, I remember it, maybe 50 years later. So that just goes to show you that it's possible to actually go through this process of re-rehearsing and be able to convert short-term memory to long-term memory. Now, how memory is formed at the molecular level, usually you have a steady state situation where you're going to have these neurotransmitters then activating receptors. It's possible when you go from short-term to long-term plasticity that you cross a threshold condition. You have enough of these neurotransmitter fluxes going across the cleft here that you actually start to synthesize new proteins. So what happens is you will have selective ion channels open up as a function of the concentration of the neurotransmitter. That will then set a chain of events happening in cell signaling that actually ultimately involves in the production of new receptors. So that's where the actual microscopic basis for long-term potentiation comes from. What's not shown in this picture, though, is what's the role of the cell membrane, the lipid bilayer that actually makes up this area, this interface right here, where all the membrane proteins and all the actions occurring here, where everything is being transduced. And the cell membrane, of course, is made up primarily of lipids, and actually, lipids make up about half of the brain's dry weight. The other being proteins and small molecules, carbohydrates, and things like this. Lipids are very important. There's a variety of different kinds of lipids, and there's also cholesterol. They're involved in membrane formation, signaling, energy storage, homeostasis maintenance. In human beings, it's estimated there's over 100,000 different kinds of lipids. Whereas in model systems like the one I'm going to be talking about, we usually talk about just one single kind of lipids. So there's a lot of complexity there that we are only beginning to address. Brain lipids are important targets for a variety of different diseases and treatment of diseases. And interestingly, I found recently that, for Alzheimer's, usually the conventional wisdom is that you have these amyloid plaques that build up, but there's now also evidence that lipids and changes to the lipidome may also be important for Alzheimer's. Now, the model of the plasma membrane and the role of lipids has evolved over the past 100 years. So basically, in 1920s, you would have a model for the cell membrane just being this self-assembly lipid bilayer into two leaflets, separated by a little gap here. A little bit later in the 30s, it became known that membrane proteins were important. And the model was that they basically formed an overlayer on top of the bilayer on both sides. In 1972, you had the fluid mosaic level model, where you have the proteins moving around, diffusing fluidly in the membrane, along with other kinds of biomolecules like carbohydrates and cholesterol. And even more recently, late 90s, it really became clear how important lipid and lipid protein interactions are. So the actual, you can actually have the proteins mechanically act on the membrane, You can have proteins chemically modulate the membrane and so forth. And you can have the lipid membrane, of course, act on the proteins. Now, the model system that we're using is called a droplet interface bilayer, or DIB. And I'll frequently use the acronym DIB in my talk. And basically what it is two aqueous droplets, each one coated with a monolayer of lipid molecules, and they're positioned on these electrodes here. These are silver electrodes, about 75 microns wide. And they allow us to use micromanipulators to bring these two droplets together where they form spontaneously a lipid bilayer. And the key component here is that the oil in between the separated droplets, once you form this contact, you get a very large adhesion force because you're excluding the oil out from in between the two leaflets on the opposed monolayers. And you get a large entropically driven depletion force that brings these two droplets together. You can tune that by the molecular weight of your oil. So if you have a light oil like decane, you actually don't have a whole lot of exclusion of the oil when the bilayer forms, and you get kind of these sparse contacts here. Whereas for a higher molecular weight oil like hexadecane, you see there's much more surface area involved and much more adhesive forces involved in the depletion interactions. This will be important in my talk later on. Droplet interface bilayers are very convenient because they're really RC circuits that model biological processes at the cell membrane. You have resistance, or the membrane permeability and ion transport across a bilayer. You also have access to capacitance, proportional to the bilayer area and the thickness. And these two parameters in particular are dependent on voltage. Our experimental setup is we have what's known as a patch clamp rig on a confocal microscope. We have a couple of these rigs now. And what you're seeing here, this is a confocal turf microscope where you can do a total internal reflectance fluorescence. And you see the two electrodes coming in here from the side. And we're actually in this system here, we're able to make patch clamp. As you see here, we're actually be able to isolate single proteins in a giant unilamellar vesicle, a GUV. So we have the ability to do electrophysiology concurrent with single molecule fluorescence and fast bright field imaging to look at connections between membrane fluctuation characterization and protein conduction. Now, what really got us started in this field is we wanted to be able to look at the difference between natural and synthetic sensory nervous systems. We wanted to be able to get an idea of how the brain does computation and use that to inform us how to make neuromorphic devices. So most neuromorphic devices are in the solid state. They're usually tunnel junctions. But the tunnel junction concept is very different than how computation is actually done in the brain, right? Communication in the brain is ionic, not electronic. And we're dealing with concentration thresholds of neurotransmitters and so forth. Whereas in the solid state memristors and other kinds of neuromorphic elements, we're talking about tunnel junctions and tunneling currents. But basically what's happening here in this picture is that you have a lot of these two-terminal memristors in between these wires here, and they can be mapped onto a neural network. And this is how, at an abstract layer, how the brain does computation. So we want to be able to kind of merge these two and come up with a hybrid device paradigm where you can have soft matter make artificial neuromorphic elements and networks. Now, memristor is a portmanteau meaning memory resistor. Is a circuit element that remembers. And basically what it means is that the conductivity of a certain memristor is going to depend on its past history. So if you have a higher current in this example right here on the left, that means that you're going to have a lower resistance when you measure it again. So it's very similar to how neurons connect. So if you have a neuron that has a lot of synaptic connections, the connections between these neurons tend to get stronger with time. And then the converse is also true, that if it has a few connections that's weak, that it gets weaker with time. So you have this concept known as Hebbian plasticity, neurons that wire together, fire together. Now we made the first soft matter kind of memristor from droplet interface bilayers back in 2018, where we used very simple ion channels based on alamethicin peptides. These are antimicrobial peptides that insert and form these pores, conductive pores in the membrane above a certain threshold voltage. And you can see that here in the IV curves. What you're seeing here is that there's zero current until you reach a threshold, and then you see the hysteresis loop on both sides as you scan plus and minus voltage. This hysteresis loop is known as the smoking gun signature of a memristor. And what's happening here, sorry, is that at the voltage threshold, you drive these monomers to assemble into this conducting pore. Now, the necessary conditions to say that you have a memristor is, of course, the pinch hysteresis. The pinch hysteresis loop must decrease with frequency. And you can see that here as you're going to slower and slower scan rates. And in the limit of frequency going to infinity, it should collapse to a single valued function. And the memristors in the soft matter systems can be very, very sensitive to environmental conditions. So here we have the same memristor. This is DPHPC lipids. It's a certain lipid that's very fluid. It's a liquid crystal. And even at 50 degrees, it's still a liquid crystalline phase. But you see that the pinch hysteresis and the hysteresis loops have changed as a function of temperature in a very dramatic way. And if we change the lipids to now going from a single composition lipid to what's known as brain total lipid extract, you can buy this from Avanti. It's all the lipids from a pig brain. It's a very different response. Basically, what's happening here in terms of the physics is that you have a time lag between the pore relaxation rate and the steady state number of pores, both of which are voltage dependent. And I'll go into more detail about how that works. Nice thing about memristors and also what happens in the brain is that they exhibit short-term synaptic plasticity. So plasticity means that, very similar to the idea of memristance, the activity of a synapse is going to depend on how much it's used. And so you can look at very simple models for synaptic plasticity known as paired pulse facilitation and paired pulse depression. And for paired pulse facilitation, what that means is that if you have two electrical pulses or two stimuli to a neuron or a synapse close enough in time relative to the relaxation time, the signal will continuously increase as you have more and more pulses, and you see that here. If we increase the interval between pulses, so now it's longer than the relaxation rate of the ion channels, then you get PPD or paired pulse depression. The STP, the short-term plasticity, is very sensitive to environmental conditions. Here we see that at 50 degrees C, the lipid membrane first exhibits PPF, but then as more and more oil goes into the membrane at the higher temperature, you get PPD because the membrane gets thicker, and then the ion channels are no longer able to form. And then, of course, if we go to 10 milliseconds, PPD accelerates. So we can map out the normalized current, that is the PPF response as a function of interval here. And you see that as you go from 10 milliseconds to 1.5 seconds, that it really goes from facilitation to depression. Short-term plasticity is volatile, so it's not really good for setting a bit. What it is good for, though, is cell signaling. So it has a lot of functionalities that are important for signaling, such as frequency filtering. You see that here on the left. You can have adaptation sensitization interactions. So you can have the ability to be either highly sensitized to a signal or the ability to be able to ignore it after you've already been able to detect it, and you have gain control. We later showed that it's also possible to have memory capacitance in lipid bilayer membranes. In this case, there's no ion channels, it's just the lipids. And so what you're seeing here on the movie on the right is, as a function of voltage, there are two things happening. One is that you're driving the oil residual oil out of the hydrophobic core of the membrane. And as you're doing that, you're also increasing the charge. You're charging up the capacitor here, and so you see that happening as a function of voltage, kind of cycling in this video here. What's happening is that there's two things that depend on voltage. One is known as electrowetting, where you're increasing the area of the bilayer. And that's because you're changing the interfacial tension of the bilayer and the contact angle. So there you're not actually doing anything to the bilayer itself, you're just making more of it. And then you have electrocompression. So what that happens is that there's electrical stress, attractive stress that brings the two leaflets closer together, so it squishes them. Both are dependent on voltage. And so what you're able to generate are pinch hysteresis, but in this case, in terms of the charge. So you're looking at charge versus voltage here, and you also have this pinch hysteresis. And you can convert that to capacitance, and you can see as a function of frequency, you have different capacitance responses and different kinds of pinch hysteresis in the charge voltage plane. What you're seeing here in the insets is that as you're doing this, you're also dissipating energy. That's what the slow rise in the energy is. Droplet interface bilayers exhibit STP in capacitance too. So what you're seeing here is paired pulse facilitation for decane, a smaller oil, lighter weight oil, and hexadecane. You see that the decane facilitation is much more pronounced because you're more efficient at driving out the oil from the membrane as a function of voltage. And that brings the membrane closer together and increases the capacitance. One nice thing about DIBs is that you could actually prepare asymmetric membranes very easily just by having different droplets before you bring them together. And so what you see here is if we switch out one of the leaflets, then we can get even more functionality in terms of the STP traces that we can show here. Long-term potentiation then would be something that lasts much longer than this volatile STP. So it's synaptic connections between neurons that become stronger with activation, but it's persistent. So once it happens, it lasts kind of like that example I described earlier about the yellow house that I tried to memorize. What you're seeing here is that you get this increase in the graded potential. You have more receptors at the synaptic cleft, and then your stimulus and your neuronal response lasts longer and is stronger. The question here, though, is what roles do lipids play in this? Like I said before at the very beginning of the talk, we do not have the same scaling of the ratio of the number of ion channels to body size, which suggests that lipid membranes have some role too. The expression and localization of receptors and ion channels is very energy intensive. It takes a lot of energy to make these things. And so if you can somehow make the whole process more efficient by really, taking advantage of lipid protein interactions, you'll be able to have the same kind of functionality, but without having to increase the number of ion channels. First observation of LTP, of long-term potentiation, was done in a rabbit's brain in 1973 by Bliss and Lermeux. And basically what they're doing is that they're stimulating and recording from an area in the limbic system of the brain that's very close to the hippocampus. And so what you're seeing here is the recording the activity of the synapses here after being stimulated and induced into LTP with and without the cell body. So without just looking at either the synapses or actually the neurons, their cell bodies and the synapses together. And what you see is that this control experiment tells us that LTP really in the brain really depends on expression of proteins. Protein expression is definitely required. And again, this is an energy intensive process. So this is a significant finding about how and why LTP is transduced and expressed and can be detected. It's very dependent on long-lived protein expression. We recently demonstrated long potentiation, but without ion channels at all. So what we did was we used the capacitance of a lipid-only membrane in a DIB to demonstrate LTP. So what you're seeing here, this is the same pinch hysteresis between charge and voltage that I showed earlier. You can integrate the hysteresis loop on both lobes here in the IV curves, and that will give you the capacitive energy. capacitive energy is the integral of these two lobes here. And what we see is that if we do the following experiment, so if we apply a sinusoidal voltage for 1 hour, and then after that, a series of just very short measurements, what we see is that during this hour, we have pretty much a steady state response, the stored energy here. But then when we take off that constant energy, that constant voltage, and record the energy, then it jumps way up like a factor of 2, and it stays high all the way out here until like 17 hours. Eventually, it does relax back down to the initial ground state, but this is a very long-lived effect. We see it both for one lobe first or the other lobe. So if we apply positive going voltage and then negative going voltage, we see similar kind of behaviors, although quantitatively they're a little bit different. It is possible to erase it too. So if we're having a positive going sine wave, for example, and then we immediately switch the polarity, we quickly erase the response. Now, to try to get a better understanding of this and how capacitance factors in, it's useful to look at the difference between capacitors and memcapacitors. And one has memory and the other does not. The standard capacitor is just a dielectric between two electrodes. And when you look at the charge voltage plane, what you have is basically a straight line. The slope of this straight line is the capacitance, and the shaded area underneath it is the energy stored in the electric field between the two plates in that capacitor. And capacitance is just charge versus volts. What we're seeing here in the fingerprint of a memcapacitive system is different in that right now, what we're doing here is we're looking at the differential capacitance. That is not Q equals CV, but dQ dV. And that's important. That distinction is important for how a memcapacitor gives rise to LTP, whereas a capacitor does not. So, like I said before, it's the differential capacitance that matters in DIBS. Here I'm showing the same movie again. This is the dissipation of the energy as a function of time. And so what these higher oscillation frequencies are, these are the static capacitance. This is capacitance equals Q over V. And as we're going through the sine wave, we're just increasing and decreasing the capacitance and also the energy. Compare that with the slow long-term change in the slope of this trace here. This is the differential nonlinear capacitance. So now instead of Q over V, it's dQ over dV. And if we do the calculus, it's C plus V dC dV, which we can integrate, and we see that the energy going into the system is going to be dependent on this integral right here. And importantly, you have this term, this extra term right here. That's going to be important. This is known as the dissipation of the energy. If we look at an equivalent RC circuit, we have the seat of EMF, a battery or a voltage. There's a capacitor and then a resistor in series. We can look at the different components here. This is what's called the dissipation. You can think of this as the heat being generated as the current is going around this circuit and is being dissipated in the resistor here. The actual energy of the membrane is these two terms right here, and this is the dissipation. And that's shown here. Importantly, in order to be able to store memory in your membrane as a capacitive energy term, you have to have the system pumped into a non-equilibrium steady state. You have to continually supply energy to the system, and you're going to dissipate some of that energy as heat. So a non-equilibrium steady state in general is a state in a system where external forces maintain a constant imbalance, result in properties that do not change steady state over time despite being away from thermal equilibrium. And that's what's happening here. And that's how we're able to have a memcapacitor store capacitive energy. We also explored different ions. So the first ion that we chose was potassium, which works the best, incidentally. What you're seeing here are the different parts of the energy expression of the equivalent circuit that I was describing earlier. This is the total energy. This is the dissipation component. And what you're seeing here is a function of ions, potassium, sodium, lithium, and then thulium, which is a trivalent large cation, that the response just kind of like disappears as you're going to these different ions. So potassium is really the best candidate for seeing this effect. To get an idea for why that would be the case, what we're showing here are this is the series of charge voltage loops as we go through the training period. It starts out as black line, and then as we're doing the training, it's going to be replaced by red lines, and then after training, blue. So what you're seeing here in both the charge voltage plane and also in capacitive voltage squared, which is showing the dissipation energy component, that you have an initial capacitance loop. The capacitance drops as a function of training, and then when the training is turned off, it springs back a little bit. So you go from the red to the blue. If we look at the capacitance, the energy stored in the capacitor while we're doing this process, what we see is that if we compare the black trace here for the capacitance versus the blue one down here, the black one corresponds to the beginning of the experiment when we're training the membrane, but then the blue one after the training is ended shows that the capacitance dropped, but the amount of energy stored inside there, the loop here area, and this hysteresis loop, is increased. This is our signature of how the memcapacitor is able to store energy and function as a LTP element. What's happening at a molecular level, which has been informed by molecular dynamics simulations, is that you're getting charge density variations in the leaflet radii curvature as you're training the membrane. These lipids, they're phosphatidylcholine lipids, deuterionic. They're flexoelectric and piezoelectric. Flexoelectric meaning that voltages couple to membrane bending, and piezo means that they couple to the membrane thickness. Both of these contribute to the energy dissipation in a non-equilibrium steady state. And they have two parts to it. There's the dipole potential and a surface potential that make up the transmembrane potential. And what you're seeing here is these fluctuations in the membrane curvature as we're cycling, as we're training the membrane. The charges are screened because we're at high salt concentrations, and so we have Debye screening. So what we're really seeing is changes in the dipole potential. What happens, and this has been informed by molecular dynamics simulations, is that you get an asymmetry in the polarization as you train the membrane and you drive it in its non-equilibrium steady state. So what does that mean at a molecular level? Well, what you're seeing here is changes in interfacial dipole potentials and the ordering of water at the membrane interfaces. If you look at the like a cartoon of a lipid molecule, for a PC lipid, you've got the choline group and the phosphate, so that makes this zwitterionic neutral but highly polarizable head group. And then you have the carbonyl oxygens further embedded near the glycerol groups. Now, water molecules and ions are going to interact with this charge field here. And so we see that as we continue to cycle the device, we are going to affect how the water molecules interact with these head groups. What happens is that as we are driving this thing, as we're training it, we get a huge net orientational polarization of the water molecule dipoles in the stern layer that is right within 3 angstroms of the head groups. And it's more, the effect, this polarization effect, this orientational polarization is most pronounced for potassium. It just happens that the potassium has the right hydration energies and polarizability and so forth, that it makes the most ordered polarization dipole here at the interface. Sodium and lithium are of course not as good, and thulium is terrible because it's very hydrophilic, so it really disrupts the head group fusion here. Now, recently, we've been trying to see, is it possible to couple LTP and capacitance with ion channels? And to start that discussion, we went to gramicidin. Gramicidin is a very simple conductive pore. it forms what's known as electrical synapses. Now, alamethicin and most ion channels are known as chemical synapses in that they transduce chemical to electrical signals. Whereas electrical synapses like gramicidin form more like gap junctions, like wires that just go across the membrane here. And the way that the gramicidin forms, the dimer forms, is that you have these two monomers that are freely diffusing in both leaflets. And when they happen to line up with each other, and then the membrane adjusts its curvature and compression in order to form a dimer, and then you have conduction. And just as before, we were able to show STP. So this is actually the work of Andy Sarles. What you're seeing here is that as a function of PPF, you get this increase in current, and then when you increase the separation between pulses, it drops and you get depression. What you're seeing here is this. I can play it. I'm not sure I can play this while I'm also having the. There we go. So what you're seeing here is the changing of the bilayer area as a function of voltage. So these are the two electrodes. This is the water droplets. Here's the oil out here, and this is the interface right in between them. And what I'm doing here is I'm advancing the slide. I'm going through PPF. So ignore all this spike stuff from electrical noise. But what you're seeing here is that the current is increasing as a function of time when the pulses are close together, a 5 millisecond interval between pulses. And while you're doing that, the area is increasing. So the reason why the area that is given by this diameter of the bilayer here is increasing is because you have electrowetting. So you're able to form more bilayer because you're changing the contact angle between the droplets as a function of voltage. And then you're able to do that. And then eventually we switched to PPD. So now we see depression, and now the area is dropping. So if I go through this PPF to PPD interface here, you see the area increase and then drop and then decrease. So that's what I'm trying to show here. Hopefully you can see that. Okay, so go to the next slide. Turn on my laser pointer again. All right. So if I actually plot that, both the area, this is the red trace here, and the current. So the current came from the current that you saw on the right-hand side of the previous slide. And area comes from the images of the bilayer droplets on the left-hand side of the last slide. We see that both area and current increase as single exponentials. They have single exponential timescales. And then when we go from PPF to PPD, it decays with a single exponential. Compare that with a mixture of hexadecane with a little bit of dodecane, so a little bit of smaller molecular weight oil in there. And now the area increase is still a single exponential, but the current increase is a double exponential. And you see that it increases much higher, much faster than just the area. So what we see here is that it's not just electrowetting that's responsible for the PPF, as in the case for pure hexadecane. But now with a little bit of the smaller molecular weight oil, we are able to have much higher increase in current. That's not due to just formation of new ion channels, formation of new membrane area and getting new ion channels for that reason. We're actually increasing the efficiency of the ion channels that are there. If we look at normalized traces for PPF on top right and PPD on the bottom right, we see that the timescales for the pure C16, the hexadecane, is we get one timescale of about 5 seconds. Whereas for the mixture, we have two timescales, 3 seconds and 23 seconds. And then likewise for PPD, we have one decay at 5.6 seconds for pure hexadecane, and then two timescales, 3 seconds again, and then 100 seconds for the second component to that. And so what's happening here? What's happening is that you are looking at the effect of having that little bit of dodecane in the mixture go in and out of the membrane and modulate its efficiency for making the gramicidin more conductive. Here we're showing PPF traces as a function of interval for both PPF and PPD. So for PPF, the 5 millisecond intervals, of course, give you the biggest effect, the biggest amount of STP. And then it drops as a function of interval between the pulses. And then for PPD, of course, we start at 15, and then as we go longer in time, we get more of a depression, so more of an effect at longer times. If we compare that with what's happening in the mixture, we see that the mixture has a very different kind of response, is much higher, and the time scales are different. Now we have a bi-exponential in both PPF and PPD. Very similar to what I just showed earlier, or the case of the video that I showed. So what's happening here is that you have two different scenarios of STP. You have the one that's happening in hexadecane, driven primarily by electrowetting, and then you have the one that's also now has electrocompression in addition to electrowetting. This is a structural change to the bilayer that's affecting the efficiency of gramicidin conduction. So we're having LTP between these two STP scenarios here, and LTD for the bottom case. Our previous paper on the memcapacitor gives us some clues as to what's happening. So what I want to first have you look at the table here. These are damping and stiffness parameters for pure decane and pure hexadecane going into the bilayer when we're looking at the memcapacitor back in 2019. And so what we're able to do is basically from be able to derive coupled nonlinear differential equations that gives us the time scales for electrowetting and electrocompression. For the pure hexadecane case, everything is electrowetting. There's no real electrocompression because the hexadecane is largely excluded from the membrane because of its size. For pure decane, we have both. So now we have a large electrocompression component as well, and it has its own tau electrocompression timescales that are at work. That gives rise to the very different behavior for decane versus hexadecane. Now, in terms of gramicidin, though, decane is terrible because it forms these lenses in between the leaflets, and it makes the thickness of the membrane too large in order to be able to support conduction. But if we have instead a case where it's mostly hexadecane, but there's a little bit of dodecane in there, we get different behavior. Again, this is what I showed earlier, where we looked at the fitting, the normalized fitting of the PPF and PPD responses for pure hexane versus the mixture. We see the very different kinds of trajectories, and you can kind of make sense, right? Because here, electrowetting is faster for hexadecane because it's more easily excluded from the membrane, whereas for the mixture, you have some electrocompression going on as well, and the dodecane component is harder to drive out, and so it's going to be a little bit slower. But overall, it becomes more conductive. And then for the PPD case, you get the converse is basically true. Now you turn off the voltage, and now you're waiting for the oil to come back in, and it takes a long time for that to happen. So what's happening here is, this is like a figure from a paper recently published. What they did was they had they added a little bit of a different kind of amplifier in here, and that affected the formation of the dimer. So we're doing something very similar, but we're adding oil inside the membrane here in the DIBs, but we're able to change the way that the dimer forms. And what we're doing here is that we're using electric fields, electric compression that increases the degree of hydrophobic match. That is the matching between the hydrophobic acyl chains, the lipids, and the hydrophobic residues of the peptide. Molecular dopates, in this case amphiphils, but in our case oil, alters the fluidity, the bending stiffness, and the area per molecule to either enhance or degrade hydrophobic match. Higher concentrations of the lighter oil, in our case dodecane, degrade hydrophobic match by thickening the membrane. So we have a case where if we have hexadecane, that was what we thought was the best case, but then we add a little bit of lighter oil, it gets even better. But too much of that, and it gets bad because we've added too much of the lighter component. We formed oil lenses in between the leaflets, and we screwed up our hydrophobic match here. The same kind of behavior is seen in volatile anesthetics. They have very similar behavior to alkanes. And so what you're seeing here is this is the Meyer-Overton curve. It's the degree of partitioning of these different volatile anesthetics in the membrane, and how they affect the bending modulus. And so as we go from a relatively low partition coefficient for ethylene to this compound out here, methoxyfluorane, the partition is much higher, and we see a much larger change in how the bending modulus is affected, and that is going to affect your hydrophobic match to your peptide. So what happens is when you have these volatile anesthetics, the membrane stiffness goes down, the bilayer thickness goes down, and the area per lipid goes up. Now, anesthetics frequently target specific ion channels, but lipid bilayer structural alterations are also important and also believed to facilitate anesthetic action. So what could be happening is that in addition to, specifically targeting a particular protein receptor, anesthetic can also change the efficiency for how that receptor works in the membrane by altering membrane protein interactions. What we're looking at here is an SDP LTP energy landscape at synapses. Now, for actual synapse in biology, we can look at what's happening in terms of the calcium pools. The calcium is very important for signaling because it actually triggers through a series of cascades the synthesis of new receptors and new other biomolecules that are involved in signaling. And so you can actually look at the PPF and PPD as a function of interval time and come up with a model for the kinetics. So you can have a readily releasable pool of calcium present in endosomes, vesicles very close to the membrane. And you can also have access to a reserve pool for calcium and other kinds of receptors and other important things coming from the mitochondria that are then accessed during LTP. So you have SDP occurring right there at the membrane, and you have LTP due to triggering of migration and synthesis of important biomolecules. So in an energy landscape, you can think of LTP and LTD being transitions between these different minima on your energy versus reaction coordinate landscape. And then the STP would be metastable transitions occurring over these and down these barriers and into the wells here. And that's just what we see, I believe, for how the membrane couples to gramicidin. So if we look at this reaction coordinate, where we have the two monomers, they're separated from the dimer by this barrier. The barrier is believed to, the actual transition state is believed to have a separation distance of about 1.6 angstroms. The dimer length is about 2.2 nanometers, whereas the bilayer is about 3.6 to 4.4 nanometers. And so what's happening is that you're getting this compression and the bending of the bilayer that brings the system over the barrier and into this dimer state. If you have this entropically driven depletion force that you're having in DIBS when you form the bilayer and you're driving the oil out, you create your system in a metastable state until the oil can go back into the membrane when the voltage is turned off and then be able to separate these two monomers who form the dimer. What happens is you go from pure hexadecane to the dodecane hexadecane mixes, you're actually lowering both the barrier and also the thermodynamic equilibrium state, delta G naught, because you're changing the structure of the bilayer. So the actual change of the structure of the bilayer is what's giving you LTP, and then the interactions that you see in peripulse facilitation and depression is giving you STP, and we're able to couple the memcapacitance of the bilayer to what's happening in terms of LTP at a molecular level. And that's what you're seeing here. STP would then be, this is what we're doing when we're applying pulses and we're sending monomers into dimers and back and forth. Whereas LTP would be the actual structural rearrangement of the lipid when we change the oil composition. Now, this is a very, artificial model system in that we're changing the oil in the DIBS setup, but there are biological parallels in terms of lipid biosynthesis and droplet lipids, lipid droplets that are formed in the mitochondria that are believed to be important for signaling. So in the last couple minutes, I want to talk about some more recent work that we've been doing. We recently had an LDRD, which is this I don't know if you have something similar, where we get in-house support for seed kind of projects. What we want to do is we want to make a DIBS, but we want to be able to look at the DIBS interface with neutrons. And I believe that it could also be possible to use this kind of idea for looking at it with X-rays. But the idea is that in order to see these kind of effects that I've been talking about today, you really need to be able to have oil cycling in and out of the membrane. Hopefully I'll be able to show you the importance of that. And so what our idea is to try to see if we can make an engineered emulsion that will allow us to have oil cycling inside and outside of the bilayer as a function of voltage. And it can be as engineered as actually making monodispersed droplets in a microfluidic droplet generator, or it could be something more elementary, but we have these capabilities in order to be able to do this. And so we're just starting an LDRD on looking at that with neutrons in the liquid reflectometer experiment beamline. So, this is probably preaching to the choir here, but we're going to be able to use the neutrons, in particular, the ability to have contrast by changing the isotopomers to look at, to really get an idea of how the systems are structured. And what would be really nice is if we could actually look at simultaneous electrophysiology and structural information with neutrons, or or perhaps with X-rays, and be able to actually, in real time, couple structural measurements with functionality. Also with Ben Doty, we've been doing some nonlinear optical studies of these systems. And what's nice about this technique is that it allows us to really selectively look only at the interfaces. So we can see a lot of the kind of phenomena that we talked about from an electrochemical point of view. We can see it structurally with looking at SFG as well. The benefits of SFG, in addition to the elimination of the bulk response, is that it allows us to look at coherent processes. And that's what's going to be important when we're talking about moving things out of equilibrium. When we're talking about non-equilibrium steady states and so forth, then we're going to be interested in that. And of course, it has very close ties to simulation. The idea here is to, with these experiments on these solid state versions of DIBS, kind of look at stroboscopic experiments where we're looking at dynamics stroboscopically, looking at different time points. The idea is to synchronize, pulse DC fields with neutron data collection and SFG in order to be able to, instead of looking at dynamics continuously, looking at it in terms of a strobe, and then be able to get these complementary measurements with millisecond time resolution of the ordering and what's happening in terms of the structure of the bilayer with conduction of the peptides and different kinds of ion channels. Of course, molecular dynamics is very important for this. We do both atomistic and also coarse-grained molecular dynamics, and these have really helped to guide and give insight into the SFG and the neutron reflectometry data, as well as the conductivity measurements with DIBS. So to close, I want to thank my collaborators, in particular, John Katsaris, of course, Ben Doty. We also have some very good and had some very good postdocs, Hayden Scott, Dima Bomatov. We work with a very bright student, Peter Podar. For our molecular dynamics, we have Jan Michael Carrillo. And this is for, Ralph Liddick. He's a professor of psychiatry at UT, at University of Tennessee. And so with that, we are starting to gear up to do actual in vivo experiments with rats, looking at see if we can use ways of changing lipid bilayer or the lipidome of rats and see how that affects memory. And with that, oh, sorry, this is Eric Watkins. He is a beam scientist at the SNS, and he's the one that's come up with this LDRD for looking at the DIBS systems that are on an interface. And with that, I want to thank you again, and I'll take any questions.
Thanks, Pat, for